Okay, very well. So if you responded in the chat, great. So in the last class, talked about gel electrophoresis, making a recombinant DNA. And we talk about restriction enzymes, right? So a quick summary, please. Who will volunteer for a quick summary? Madiha?
Sir, I was not there for the last class on Sunday. I was there for Saturday.
You were there for Saturday, where we did the. Were you there?
Yes, I was there for this palindromic sequence, not the other.
Okay, not this one. Okay, then Saifullah.
No, sir.
Saifullah, would you like to give a quick summary?
Yeah, okay, sir.
Yeah, great. Thank you. So let's begin.
This is the process for making recombinant DNA, in which a foreign DNA is ligated to a plasmid of a vector host. In this process, we use restriction enzymes, which are the enzymes that cut the DNA specific sequences. We use the same restriction enzyme for both foreign DNA and the vector DNA, so that they could cut in the same sequence.
Yes, great. It's important to use the same restriction enzyme, correct?
Then the foreign DNA is introduced to the vector DNA by heat stroke or other methods.
By heat shock, not the heat stroke, heat shock method. Heat shock and treating them with divalent cations. Mostly we use calcium 2+.
Yes, sir.
Yeah.
And when the foreign DNA enters through the pores of the membrane, it gets ligated with the vector DNA, thus forming a recombinant plasmid of DNA, which during cell division divides, and the recombinant DNA passes to the generations.
Right. Then I think we started with gel electrophoresis, right?
Yes.
Yep. So what is gel electrophoresis?
It's a process to separate different weights and strands of DNA from each other. It is used by passing electricity to a...
Before passing electricity, we need to make that gel. So it has two things. Gel electrophoresis. So first component is gel. Second is when we bring in the electricity component, and phoresis, like to separate them. So what was that gel, which is here?
Agarose.
Yes, agarose gel. What is agarose?
The natural polymer that's extracted from seaweeds.
Yes, and what is the specialty about this polymer that we can use it?
It provides sieving effects.
Yes, how? How do you think it does the sieving? Remember we made these, what is this 1x and 5x and these lines?
So the larger DNA separates from the smaller DNA.
Yeah, but what is this? What are these black and the blue things? This black is the agarose polymer. So it's based on its concentration, we can make a gel of agarose. If we are using less concentration of the agarose, we will make a gel which have bigger pores, right? Depending on what concentration fits our purpose the best, what fragments of DNA we want to separate, we will figure out one concentration, right? So higher the concentration, smaller the pores which are available. for DNA to move through it. This is what I have tried to depict here. Remember, Saifullah?
Yes, sir.
Yeah, very good. Yes, then, so gel part is clear. Now let's come to the electrophoresis part.
Electricity passed through the gel, in which, since
why are we choosing
DNA
electricity here? Why are we
says
passing?
because DNA is negatively charged, so by passing electricity, the negative particles will tend to move to the positive terminal. So in this way, we can separate the two DNAs.
Yes, correct. And then what another thing, how will they get separated on the basis of their, because charge is same, like all the DNA is negatively charged. But how will different fragments of DNA, which we have cut through restriction enzyme, will get separated?
Based on their length and weight, sir.
Yes, based on their size, right, in their length. So which one will, so which is this one?
The smaller fragments will move faster than the larger ones.
Will move faster than the larger ones, correct. Great. And how do we, I also told you, how do we visualize them, right?
Yes,
So
we use ethidium bromide to see the strands.
Yes, and but ethidium bromide is a carcinogenic agent. Basically, it goes and binds to the minor groove of the DNA, and that's how, when you see ethidium bromide, you are basically seeing where your DNA is, right? So this ethidium bromide is mixed with the DNA in this gel, in this well here, when we put these DNA samples. Okay, now then we went on to do what are the features that has to be present in every vector DNA, which is every plasmid vector, to facilitate the process of gene cloning. So what was this? Would you like to just finish with this, Saifullah?
So for the process of cloning, DNA should have the following three characteristics.
Not the DNA, the vector, the plasmid DNA. The plasmid vector should have the following characteristics.
Yes. Origin of replication, selective markers, and cloning sites.
Yes, origin of replication, because without it you cannot replicate any piece of DNA inside the cell. Selectable marker, so that we can make sure that our plasmid has successfully been incorporated, and so our gene of interest is inside the cell, inside the host. And cloning site, of course, because if it does not have a cloning site, we cannot insert our gene of interest into a plasmid vector, correct? Very good. So I gave you homework to go back and look at agrobacterium and to read a little bit about cloning vectors. Did you go back and do that? Anyone read about cloning vectors? No? Okay, what is Agrobacterium tumefaciens? You must have studied about this. As the name suggests, Agrobacterium tumefaciens. What is it? It's a bacteria that actually is responsible for making tumors in a plant. You know which is that plant? So if you have not done it, I'll be just forwarding this homework to the next class. So this is also a homework. Please write it down, everyone. And I want to make sure that Madiha, Saifullah, and Ahmad, all three of you have written it. You have to figure it about Agrobacterium tumefaciens Cloning vector, I just taught you everything I taught you. The plasmid vector is the cloning vector. But there is something, how it is related to Agrobacterium tumefaciens, you have to figure it out. When you will read it, you will understand. It's there also in your NCERT. So have you noted this homework?
Yes, sir.
Yes, you're right, Madhya. But do you know how is it related to cloning vectors? So the answer lies in, so as I told you, it induces tumor, right? Okay. Madhya is writing something in the chat. Not completely. Okay, fine, not a problem. So you have to go back today and figure it out. So just remember that Agrobacterium tumefaciens, as the word says, it has the capability to generate tumors in the root cells of a plant. And they are pathogenic to the plant because of their tumor causing property. Okay. Now, how does this cause the tumor? So this tumor inducing also happens through a plasmid. So this bacteria, Agrobacterium tumefaciens, has its own plasmid, which is called the TI plasmid. Now the TI plasmid is called TI because it's tumor-inducing plasmid. So you have to go back and read about this a little, okay? So this is your homework. I've told you the basic foundation, which will help you doing this assignment, okay? Everyone? Is it clear?
Yes, sir.
Okay, great. So in today's class, let's finally, whatever we have studied, all of the things that we have studied are the tools. Okay. Now let's go to an overall process and start from the beginning and do a recombinant DNA technology. Let's execute a full thing. So today is November. Okay. So basically this is the steps to the process, the whole process. Steps to the processes of recombinant DNA technology. Okay, so you have to tell me now because I've taught everything to you. So all the pieces of the puzzle I have taught you. Now I have to put all the pieces in place. So suppose now you, after reading all of this, you want to do a gene editing. You want to engineer a DNA. You want to do DNA engineering or a genome engineering, and you want to create a recombinant DNA technology. You want to put it into a host organism, let's say a bacteria, and you want to get the gene product out. So where will you begin? What will you start with? What is the first thing that you need to do? What is the first thing that you would like to do, people?
So we would have to isolate the DNAs.
First, yes, correct. You first have to isolate which DNA?
And this vector DNA.
No. How can you isolate a vector DNA?
Or is the foreign DNA?
You first have to isolate the genetic material, the total DNA from an organism from which you want to get your gene of interest, right? There is no way to just get your gene of interest in, like, there are ways. But first we will isolate the genetic material of the host. Yes, Madhya. Correct. So genetic material, which is of course a DNA of not host, sorry, of the organism. Host in this case is, remember, the host is the one which is taking our plasmid DNA. So let's not call our source as host, okay? Is it clear, Madhya? I am not considering here host. The host will be the bacterial cell which will get our, because otherwise we have to say host twice and it will be confusing, okay? So this is the total DNA from the organism from which we want to take out any gene of our interest. So how do we do that? Now tell me the properties of DNA. First of all, why do we need to isolate? Write down. Why isolation of genetic material is done? Because to cut DNA with restriction enzymes, it has to be purified. It has to be in pure form, or it has to be purified. So there should not be contamination, no other macromolecules, no protein, no lipid should be there so that the RE can work. So we are isolating the genetic material because without this we cannot proceed. Now, what is the problem in isolating DNA? Tell me. First problem is that it is inside a eukaryotic cell. It is very much enclosed, right? Am I clear? So Mehraj and Mamuna has joined. Good evening, Mehraj and Mamuna. Am I audible to both of you?
Yes, sir.
Yeah. Okay. So we are doing, we are finally putting all the pieces of the puzzle together, and we are going through the whole process of recombinant DNA technology, starting from the beginning. Okay. And the first step is isolating the genetic material, the DNA, from the source from which we want our gene of interest to be extracted. Okay. So if you want to take a gene of interest from human, you have to extract DNA from a human cell, cheek cell or something. If you want to do it from a plant, you have to do it from a plant cell or any organism. But whatever source you choose, what is the first thing that you need to do? You have to get through some membranes, because DNA is sitting very safely inside multiple membranes, right? Am I correct? So the first thing is to break open the cell membrane, right? And not only cell membranes, but what else? Plasma membrane and also the nuclear membrane. But since both these membranes are biologically very similar, they are lipid bilayers. So can you tell me how can we break open?
So we can use lipase.
We can use
Lipase for digesting lipids.
lipase, yes. Or, so where we'll get these enzymes? Suppose we are taking it from a plant cell. Lipase won't work, right? Because it has a cell wall before the membrane. Then what will you use? Or suppose I'm interested in a gene from a fungus, let's say yeast, Saccharomyces. Then what will you use? For plant, we will have to treat them with cellulase, right? So we have to use enzymes like cellulase for plant cell because it has a cellulosic cell wall made up of cellulose. We have to use chitinase. Remember, the cell wall of fungi contains chitin. Chitin is the same biopolymer which is also present in the exoskeleton of arthropods. So it's very strong. We have to use chitinase. The word ase means enzyme which degrades it. For fungi, yeast. And in general, what we can use? So you said lipase. Apart from lipase, we also need to break down certain proteins and things like that. So is there any one source where I can get all of this? All the enzymes that degrade a cell, where can I find it? I can use the enzyme called lysozyme. It lyses the bacterial cell. So if I'm working with bacteria, I can use lysozyme. If I'm working with plants, I have to use cellulase, chitinase, etc. So once you use any of these enzymes, basically what you are doing is you are releasing the DNA. But you are not just releasing DNA. You are releasing DNA along with all the macromolecules. Which are present inside the cell, right? Now it contains proteins, it contains RNA, it contains lipids. So now what you can do is, now you can treat that whole extract with protease, okay, with ribonuclease. So ribonuclease will remove RNA because we don't want RNA to come into our extraction. We just want DNA. So we will use ribonuclease. This is now the cell extract. So on this cell extract, we will treat it with ribonuclease to get rid of RNA. Then we'll treat it with protease to get rid of proteins. You also said lipase, right? Get rid of lipids and fats. And what else? What is left? Our DNA is left. So the next step is, after this treatment is done, we will be left with DNA. But what is the property of DNA as a molecule? So DNA can actually be precipitated out in chilled alcohol. So if you use very cool chilled alcohol, which is ethanol, so you can precipitate your purified DNA in that ethanol. So what we do, we just put ethanol in that test tube from the sides of the wall. Okay, so what happens is, ethanol, it makes a layer. If you agitate it, it will mix. So we don't have to agitate it. We have to make a layer of ethanol on the top. And all your DNA, it can be precipitated using chilled ethanol. Yes, Madhya. So Madhya is saying that the DNA comes out as white threads, and those are DNA. So basically, those are not DNA as per se. Those are thousands and millions of DNA, intertwined together. These fine threads are not single DNA. Okay. So Madhya has extracted a spinach DNA recently. How was it, Madhya? Can you speak? If you would like to tell us, how did you do it? Did you follow the same protocol I am telling you?
Kind of, yes, sir.
Yes. So what did you do? You must have used some detergent, right?
Yes, we used a detergent and then
Yes.
we
Why did you use
agitated
a
and we kept
detergent?
it for a while.
Because detergent breaks open the membrane. So what do you use as a source? Fruit? Spinach. Or you said spinach already.
Yes.
So did you boil the spinach or is it raw spinach?
No, sir. We actually made a spinach solution by like adding detergent in it and then we strained it using a muslin cloth and then that solution was used in the test tube with detergent and pineapple juice.
Great. So pineapple juice has some extract. Pineapple juice was just to, so if you added pineapple juice, you also got DNA of pineapple, right? Along with spinach. So you extracted from a mixture of pineapple and spinach. So people use detergent because it breaks open the membrane, then everything comes out. Then you use muslin cloth to get out of, to sieve basically all the waste, like the bulk and the solution that was in the test tube. Then you added ethanol to it, right? Very, very finally, pouring from the sides, you must have added ethanol. So you can write down purified. Thank you so much, Madhya. So you can write purified DNA precipitates after adding chilled ethanol. Now it has to be chilled, okay?
So why are we using chilled ethanol? Can you explain again?
For what we are using, like the whole process?
Yes.
Yeah. So DNA is soluble in water, right? It's in your plasma membrane. It can easily be soluble in water. Correct? Now we are using chilled ethanol because chilled ethanol do not have its own, like when you chill any liquid, for that matter, if you chill water, the solubility of that anything for it to solubilize in it decreases. So you can do this experiment, very simple experiment, by taking a chilled water and a warm water and a normal water. Take 3 temperatures of water. Water is the same chemical. One is chilled, one is normal, one is a little warm. Then add 1 teaspoon of sugar in all and see which one dissolves faster. Which do you think dissolves faster?
And the warm one.
Exactly. Why?
Because solubility in it is decreases.
High, right? In chilled, it's decreased. So chilled ethanol gives the best precipitations of DNA. And it is done very carefully. So you tilt the tube and then you start pouring ethanol very slowly so that it just goes and sits at the top, makes a layer. If you just shake the tube, everything will get mixed and you will not get DNA. That's a failure of the experiment. Okay. Is it clear?
Yes, sir.
Okay, very well. So you can write that it can be seen as a fine thread suspension. Fine thread suspension. So we got the DNA. Great. Is there a way that I can be sure that it's DNA only? Whatever I have got. Is there a way I can just say that, okay, it's DNA only? It's not something else I got? Because I would like to be sure that it's DNA before moving forward and doing everything. And then in the end, finding that, oh, I did not get the DNA in the first place. It was something else. So one way to make sure is degrade everything else, which is there. So Madhya, did you use this step? Did you do this step where you treated the cell extract with ribonuclease, protease, lipase, amylase, etc.?
No, sir, we just extracted it after like adding ethanol and then just took it in a salt solution and viewed it under a microscope.
Okay, so mostly schools do that because these enzymes are very, very costly. So you can also
yes, it's costly. That's why we also didn't do it.
Yes. So amylase is for carbs and sugars. Okay. So these enzymes are very costly, but if you are not using it, then there are chances that your DNA, which you are getting as a fine thread, basically it might have all the contamination. It can have, remember I told you that RNA can also come out, right? So there can be RNA, some proteins, lipids, and amylase associated with that thread. So you will not get a pure DNA. But the whole point in a school experiment is to show you that, see, you can extract DNA. So they showed you that, and it makes sense because these are very, very expensive enzymes. But when you are doing it for research purposes, where you are doing some serious business of taking out a DNA, taking your gene of interest from it, and then putting it in a vector, you are doing a lot of, things which require a lot of expensive reagents. So there you want to be sure, right, that it's DNA. So you first treat and make sure that all other macromolecules are degraded. Second, you can also test it using something called DNA absorbance. So there is a machine called nanodrop where you take a very small amount of your sample and see the absorbance. So DNA, because of its structure, its double helical structure, it shows a particular absorbance at 260 nanometers of wavelength. So if you get a very clean peak showing a very clean absorbance, if it has a contamination with other proteins, carbs, RNA, etc., you will get multiple peaks. If you get one single peak, it is telling you that only one type of macromolecule is in your solution, and that is your DNA. Is it clear? So this is not in your syllabus, but I'm just telling you. You should know that when we are purifying, we have to be pure that it is. We have to be sure that it is a pure DNA. Make sense, everyone?
Yes, sir.
Yep. Okay. So what is the next step? First was to isolate the DNA. Done. We isolated. Now what do you want to do? Second step. You got the whole genome of the organism. Now what do you want to do? Nothing? How can you get stuck after the first step? Now, do we need the whole genome? Do we need the whole DNA of an organism to proceed?
We have to cut it
Yes,
at
what do
the
we need?
gene of interest.
Exactly. We have to cut it at a specific location so we can just get out the gene of interest or the DNA region that we are interested in. The one that we can put it in a vector, right? So second step is cutting the DNA at specific locations. Now, how do we do that? This we already know, because we are putting the pieces of the puzzle together in a manner, stepwise manner. We may not need to spend the same time in all the steps. So how do we do that? Simple answer is we use restriction enzymes. This is what Saifullah just summarized today, right? And the restriction enzyme that we will use here to cut our DNA, we will again use the same restriction enzyme to cut our plasmid vector, correct?
Yes, sir.
Yeah. So we will do cutting through restriction enzymes, and what we will do? We will also separate through gel electrophoresis, right? Gel electrophoresis. Great. And the same process of cutting and then isolating will be repeated for our vector DNA also. Right? So write down the next step. The process is repeated for vector DNA, which is our plasmid. And if we want to make a recombinant DNA here only at this step, what do we need to add? We need to join both the vector and our gene of interest, right? Because we have separated. So what will we add to join? Which enzyme joins?
Ligase.
Exactly. So gene of interest plus vector DNA, which is the plasmid, plus ligase. This is the enzyme that joins the DNA. If we add these three things, we have made our recombinant DNA. Okay. I also told you to go back and read about PCR. Did you go back and read about PCR? Polymerase chain reaction. So sometimes if the gene that we have isolated, if it is in enough numbers, we would like to ligate at this step only, cutting the DNA and then ligating it. It works. But sometimes we would like to, yes, Mamuna, do you have a question? Mamuna, do you have a question? No, okay. But sometimes you would like to amplify your gene of interest, okay? So this kind of amplification can also be done. Amplify means make multiple copies of the gene without putting it into the host only. So that is also possible. For that, but we cannot get it translated here. We cannot get the gene product. We cannot get the protein. We can only get the piece of that DNA replicated. So for that, there is a technique called PCR. So this step is also used, and it's mostly used because we would like to make as many recombinant DNA as possible. So we always would like to amplify our gene of interest using PCR. So amplification of gene of interest using PCR. PCR stands for polymerase. What is polymerase? The A's at the end tells you that it is an enzyme. You have studied this enzyme in DNA replication, DNA polymerase. So this is the same polymerase, but it is not a human. It's not from human source or human cells. It is a special polymerase known as Taq polymerase. I'll tell you about that, but first write polymerase chain reaction. Okay, PCR. Now, through PCR, how can we amplify? Now, what happens in our cell when DNA replicates and we make more copies? What is the first step? So this is a double-stranded DNA, let's say. Okay. This is the 5' to 3' strand. And this is the 3' to 5' strand. Now, let's say you are interested in the region from here to here. You want to amplify this region of this DNA. So what you need to do first? So this is an example of, so this DNA is called, I'm calling it dsDNA, which is double-stranded DNA, because that is the normal DNA. So first, to amplify any DNA region, so this is the region to be amplified. In cell, what happens first thing? First thing that happens is that it opens, right? There is a formation of DNA fork, right? So we would like to open the DNA. Am I correct?
Yes, sir.
So inside the cell, we know. How does it open? There is helicase that unwinds it. Then topoisomerase is also there. Another enzyme that cleaves hydrogen bond one by one, and it keeps opening the DNA strand to make a DNA fork or replication fork. But here, in our artificial conditions, how can we open? Is there any other way through which we can just break the bonds and open a DNA? Yes, we can simply heat the DNA. If you heat the DNA, DNA gets denatured. So this step from a dsDNA to this is called denaturation. Okay. So this idea was in people's head from a very long time, even before, when Carey Mullis is the person who first invented this technique of PCR. The only problem in this whole idea was, if you apply heat to open the DNA, then in that solution, you cannot use the polymerase, normal polymerase, because the human polymerase or any other mammalian polymerase, it gets degraded. It's not stable. It's not thermostable. It cannot work at very high temperatures. When I'm denaturing the DNA, denaturation happens at around 95 degrees Celsius. So this is just below the boiling point of water. So we also don't want the water to boil, because if it starts boiling, it will create pressure, vapors. Okay, which is not good for that whole experiment. So at 95, it's enough to not boil the solution in which the DNA is, and enough to just open the DNA, denature the DNA. The problem is the next step. What do we need to do next? Next step is, we would like to introduce, so this was the region that I wanted to, let's say, copy or make copies of. So I will put a primer, something that is complementary, but just a few bases of complementarity. It's called a small primer, which goes and binds to one side of our gene of interest. This is called DNA primer. The word primer you have also studied, or you must have heard in whitewashing of the walls, right? They put a primer before putting the paint, right? Primer putty. which can hold the paint. You cannot just paint the bricks. That's the same point here. You need something to initiate and initialize or start the process of DNA replication, right? So you will use primer, which will prime the whole process. So because this is a very small segment, you will add primers plus you will add DNTPs, the nucleotides, correct? Because you want to add one after the other nucleotide. But what will extend this synthesis? For that, you have to add polymerase enzyme, right? So this process, and this is the problem. So this process of adding DNA primer is known as annealing. Annealing is we are again trying to close back by binding the DNA primers to their specific position. So there will be two DNA primers. This will be called a forward primer and a reverse primer. So one primer will sit here. So which will be the forward? Remember, DNA replication only happens in one direction. What is that direction? What is one direction in which DNA can be synthesized? Sorry, come again. You went too fast. Yes, 5' to 3'. So in which of these case this extension is? So in the strand which is 5' to 3', if I'm adding a primer here, it will make it into 5' to 3' direction, right? For the other strand, it has to be 3' to 5'. So this one, I'll go to next step to show you. Next step is crucial because here we have to use DNA polymerase for the extension. But as I told you, a human DNA polymerase or a mammalian DNA polymerase is not stable at that high temperature. So for annealing, though we reduce the temperature, we can come down to as much as 72 degrees Celsius, let's say around 72 to 75 degrees Celsius, but it's still very high, right? Our normal body temperature is 37 degrees Celsius at which our DNA polymerase works. So 72 is a bit higher. It's not stable. So instead of adding a human DNA polymerase, we have extracted DNA polymerase from a seaweed, Thermus aquaticus. It's TAQ. TAQ stands for Thermus aquaticus. Anyone knows what is Thermus aquaticus? Thermus aquaticus is a bacteria which is a heat tolerant bacteria. It lives in your hot springs. It is called thermophilic bacteria. It loves to stay in hot water. Okay, so Thermus aquaticus, we have extracted DNA polymerase from this. So it's called Taq polymerase. Okay, it's stable at high temperature. So Taq polymerase plus again this DNTP. So either you can add it there or here. So let's add it here. You can also add it at that step. It's okay. So when we add DNTPs and Taq polymerase, what is going to happen? This polymerase is stable and it is going to extend the DNA synthesis. So, okay, sorry. So what was the gene of interest? Let's say this was the gene of interest that I wanted to copy. So there will be one strand here and remember semi-conservative nature of DNA replication? You're just seeing that. Is everyone understanding this? So here, our gene of interest now got 2 copies. Now, we will run this cycle multiple times. So this part here, this is adding primer to this. This is known as extension. Okay, extension happens. And then you run the same cycle. Again, this will, you go back to denaturation. So denaturation, annealing, extension, then again go back to denaturation, open it, again anneal with new primers, again do it. So we add a lot of primer in the beginning only. And now there are machines that can do PCR. They are called PCR machines, which can run as many cycles as you want. So after every cycle, your DNA will get double. The gene of interest, the copies will get double. So if you start with 1 copy, in 1 cycle, it will be 2 copies. How many copies will be there in 4 cycles?
16.
16 copies, right? So it has an exponential growth. It's just like a bacterial division. So in 30 cycles, you will get around 1 billion copies of your gene. So we run it for around, let's say 30 cycles, you will get 1 billion copies of your gene. Is it clear, everyone? So
Yes,
with
sir.
this, we can make billions of recombinant DNAs, correct? So what we need to do, we have to take the amplified fragment, and now we can use the vector and use the enzyme ligase and ligate billions of our copies to billions of vectors, and we have billions of recombinant DNA ready, correct? So write down this one last sentence, that these amplified fragments if desired, which means if you want to do this, can be used to make recombinant DNA by ligating it with the vector of plasmid. Is it clear, everyone? Everyone is done. Shall I move forward?
Yes. 1 minute, sir.
Okay. Where do you want me to stop? Here?
From the first, sir.
Here?
Yes.
Okay, let me know when you're done. Are you done, Saifullah?
1 minute.
Yeah.
So you can scroll down.
Yes. So you know that what I have marked with this red, this is the gene or region of interest in the DNA, right? Done, Saifullah?
Yes, sir.
Okay, very well. So this is done. So either you may or you may not do PCR, but mostly you should do PCR to amplify the gene of interest. And finally, you have made the recombinant DNA. What now? The product is ready. What do you want to do now? Next step, you would like to put it into the host, right? Any host cell or host organism where you want to get the product, gene product or the protein, correct?
Yes, sir.
So the next step is insertion of recombinant DNA into the host cell. If it's a bacteria, you can say host cell, or you can also say host organism because bacteria is a unicellular organism. You can also do these studies to see things in yeast or even million cell cultures, wherever you want to put this recombinant DNA and see its gene product, which is protein, and its effects. Or mostly we do it for the purpose of purifying that protein. So insertion, how we will do insertion?
Heat shock.
Yes, so basically I told you that to insert, we have to make the host cell competent, right? Heat shock or treating with divalent cations. Why are we doing all of this to a bacterial cell? to make it competent. A competent cell, did I discuss this in the last class? A competent cell is the cell which is willing now or ready to, or which accepts foreign DNA from its surrounding. Directly it can uptake the foreign DNA from the surrounding, correct? So we have to make competent in order to take up the DNA from the surrounding. And we can do this through heat shock, treating with divalent cations, which is calcium. Then it will take up the recombinant DNA. And remember, with our recombinant DNA, with our gene of interest, one more thing that we put in the plasmid. What is that one thing? Which is very important. We have to put that. Otherwise, we will not get to know that which of the cells have our DNA and which cells do not have our DNA.
As selectable markers.
Perfect. So we also have put together selectable marker. Most of the times that selectable marker is an antibiotic resistance gene. Write down a selectable marker, which is mostly an antibiotic resistance gene. It can also be something apart from that, which shows a change in the color of the metabolite or things like that. But if we are using bacterial cells, we can use an antibiotic resistant gene as a marker. Now, how does it help us? Let's say it is ampicillin resistance. Ampicillin is a antibiotic. So the gene is ampicillin resistance gene. So what we will do? We will take all these bacteria, which we have treated with heat shock, we have made it competent, then we treated it with our recombinant DNA. And we will put it on a plate. We will spread all the bacteria on a plate. We will allow them to grow for some time, of course, because we also want that our gene and our marker gene both should get expressed. So if our marker gene is getting expressed, now the bacteria has an antibiotic resistance, correct? Yes or no?
Yes, sir.
So now if I take all these bacteria and I only want to isolate those which have successfully have taken up the recombinant DNA that we created. So what I will do is I will spread this bacterial culture on a plate or a Petri plate, which is also known as an agar plate. And then I will put some antibiotic, ampicillin. So the bacteria which will have my selectable, which will have my recombinant DNA will also have the selectable marker. So we'll get the resistance and will survive. Others will die. So those who are die, we don't care about them because they didn't have our recombinant DNA. Is it clear, everyone?
Yes, sir.
So you can write down last step. The transformed cells are spread. The transformed cells are spread on agar plates These are the plates that are used to culture bacteria. And these plates containing ampicillin. So this ampicillin is already mixed with this agar when we were making the plate containing ampicillin. Now what will happen here? Write down. Only the transformants, which means those bacteria which have our gene of interest. Only the transformants will grow and form colonies. Others will die. Non-transformants will die. So a good way, right, to distinguish? Everyone done? Yes, no? Are you done, people? Shall I move forward? Anyone? No one is responding. Is it my network or? Okay. Okay. Thank you, Value. Nice. So we have inserted probably our, and if the transformants are living, then our gene of interest is being transcribed and translated inside the host cell. Now, what do we need to do? We have to grow it. We have to culture these bacteria, and we have to get this gene product, which is the protein, in very large quantities. That is why we are doing all this business. So to obtain protein products on large scale. So the fifth step, which is the industrial step, is obtaining the foreign gene product. Now we are calling it foreign because in the system from which we are obtaining it is a foreign gene. So even if it's a human gene which we have put in a bacteria, when we are isolating it, it will be called a foreign gene because we are isolating it from the bacteria, okay? Or if it is a bacterial gene, we are isolating from, let's say, yeast cell, in that case also. So what we did, we inserted. Now we are allowing, in order to get this gene product, we allow the bacteria to grow into very big, large volumes of vessels, which are called bioreactors. Okay, so we are talking about tons of liquid, so around thousands of liters, which can be processed. Okay, so write down. To produce the gene product, in large quantities, the host cells which are basically the transformants are cultured in large quantities. Okay. And for this we use bioreactors. These are large vessels, not just vessels, where we can control a lot of parameters like acidity, basicity, or the amount of oxygen that should be there. So suppose we want the log phase, so we will give, if it's aerobic bacteria, we will give more oxygen for the bacteria to grow. If it is anaerobic bacteria and we want to culture them, we will not allow oxygen to be there. So all these things, temperature, humidity, etc. So bioreactors, large vessels with controlled growth conditions. Controlled optimum growth conditions. And what are these conditions basically? Write down, these are temperature, pH, salts, oxygen, and substrate. Sometimes we also provide substrate to certain yeast or bacteria to grow on. And the protein that we are getting, because it's a protein coming from, it's a gene which is expressed in a heterologous host. Heterologous means which is not the host containing that gene naturally. So it's called a recombinant protein. Recombinant protein sometimes have to go through some post-processing modifications. So recombinant proteins are obtained when protein coding genes are expressed in heterologous hosts. For example, a human gene is expressed in a bacterial cell. Okay. So this is how we obtain. So we got lots and lots of bacteria in that big vessels. Now what to do? Now the next stage is, we don't need the bacteria. What we were interested in was just the protein, right, to begin with. So last step is called downstream processing. Once our protein of interest, which is the recombinant protein, once it is synthesized in very large quantities, we need to purify it and use it for whatever we wanted to use it for. So that is called downstream processing. Again, it's an industrial step. We cannot do it we can do it, but there's no point doing it in small quantities. So downstream processing is after the biosynthetic phase is complete, which means we have synthesized the gene. So you can write down post biosynthetic phase. Or post biosynthetic stage. The protein product, or I'm writing the gene product, which you know is protein. Yes, it's downstream. Oh, I'm so sorry. Wafia, yes, there has to be an N. I missed N. Downstream processing. Downstream processing simply means it's ready, and now what you have to do to make it a ready product so that it can be sold to the market or to the other pharmaceutical companies to make a medicine or something like that. So post biosynthetic stage, the gene product needs to be separated or isolated and purified. Isolated and purified. So this step is called protein purification. Just like we purified the DNA, there is also methods to purify protein. So they are called protein purifications. Okay. Second step is to, after purification, we have to keep it or store it in a formulation. Formulation is simply a solvent in which this protein will be stable and not will be degraded. So if it is, let's say, a receptor protein or anything which can be used to treat a disease or a disease condition, then before you make it into a drug, you have to store it. And also while making a drug, you have to keep it stable into a solution. So that solution is called formulation. So formulation is just a fancy word used for the solvent in which these products or these gene products are kept stable and safe. So this is formulation, and these are the formulations that could go under drug trials or clinical trials, right? Before it becomes any medicines. And the last step is quality control testing. You do the quality control testing, and if it passes the trials and your quality control testing, then this product is ready to be used for therapy and treatments. Is it clear, everyone? The whole process. So this is where we began with, just to summarize, we isolated the gene, the genetic material, the whole DNA, and we use restriction enzymes to cut DNA. And then before that, to break open the cell and the membrane, we use different enzymes called cellulase, chitinase, lysozymes, depending on which is the host from where we are taking the DNA. Then once the DNA is there, we would like to degrade anything else which can contaminate and only purify DNA through chilled ethanol method. Then we use restriction enzymes to cut the DNA at specific locations, which is our gene of interest. We can separate both our DNA, gene of interest, and also the vector DNA through the same process, through gel electrophoresis. And then either we can make recombinant DNA at this step, if we are happy with the number of DNA, the concentration of DNA that we have, or if the concentration of DNA is low, then we can amplify the gene of interest using PCR, polymerase chain reaction, okay, which can make billions of copies of your gene in 30 cycles, let's say. And then you can make the recombinant DNA, then you insert it into the host cell by making the host cell competent. And there also has to be a selectable marker on the basis of which you can just separate your transformants and can grow and culture it. And then once you have your transformants, you culture them on a large scale in big bioreactors, big large vessels, by providing them with optimum conditions. And then you get the recombinant protein. Okay, once you get the protein, then you purify it, isolate it, purify it, put it in a formulation where it is safe. And if you want to use it for medicines, you can go for drug trials, clinical trials, quality control testing. And finally, the product of this biotechnological process is ready to be used. Okay, so I hope this is clear to all of you. So anyone has any doubts? Maimuna, do you have any doubt? No? Is it clear? So please revise it, and you can bring your doubts in the next class if you have any. And this marks the end of this chapter. Okay. So in the next class, which is tomorrow, there will be first a test of this chapter. Okay. We'll